Okay, so on to the second step of photosynthesis. So far, what we've done is we've captured the sunlight energy, and we've basically taken that energy and we have wrapped it up or held it in ATP and NADPH molecules, but we haven't created any food. And that was kind of the whole point of photosynthesis, was to end up creating some food for the plant, and then that would eventually become food for every other creature on the planet. So to do that, we need part two, which is the Calvin cycle. And the Calvin cycle is basically it is a building process. It's an anabolic process that's going to be creating larger organic molecules using the carbon that's taken from carbon dioxide and the energy that we produced in the light reactions. So concept 10.3 says the Calvin cycle uses the chemical energy of ATP and NADPH to reduce carbon dioxide to sugar. And the Calvin cycle, much like the citric acid cycle in respiration, is called a cycle because the starting materials are recreated at the end of the cycle so that it can go over and over and over again. We're going to build sugar from smaller molecules, and we're going to use the energy from ATP to do that, and the reducing power of the electrons that are carried by that NADPH that we already created. Sometimes this is called the dark reactions. Not a great name because for the most part these reactions occur during the daylight hours because they need the energy that's created from the light reactions. The Calvin cycle doesn't go without the ATP and the NADPH from the light reactions. So at night, when the light reactions are not occurring, you're not going to get a lot of Calvin cycle occurring either. So rather than calling them dark reactions, it might be better to call them light-independent reactions because they do not require light to occur, but it does require the energy from those first light reactions. So dark reactions kind of gets people confused sometimes, so we probably don't want to use that term. While the citric acid cycle in respiration is catabolic, it is a breaking down process. The Calvin cycle is the opposite. It is anabolic. It is building carbohydrates, building larger organic molecules from the carbon dioxide, and it's consuming energy to get that done. The Calvin cycle, of course, is named after a scientist, like many of the scientific terms and things that we talk about, named after a scientist named Melvin Calvin, who studied the system back in the late 1940s and kind of helped to elucidate what the system was all about. Basically, what we have is carbon is going to enter this system as carbon dioxide, which comes from the air through the stomata of the leaves, and it's going to end up leaving the system as a molecule called G3P, glyceraldehyde 3 phosphate. And that G3P is the basis for a sugar molecule that can be created, used to create glucose and other sugars. So G3P is our final output, our final product. And for one G3P, we have to go through a fairly long process that's going to take 3 turns of this cycle, fixing 3 molecules of carbon dioxide. The Calvin cycle is divided basically into 3 portions. The first one's called carbon fixation. That's where we take the carbon from the carbon dioxide and we attach it to another molecule called fixation. And that's done by a very well-known enzyme, a very well-known protein called rubisco, which we'll talk about. The second part is called reduction, where then that molecule that was created is reduced. And then finally we have regeneration of the CO2 acceptor, which is called RUBP. That basically gets us back to where we started so the cycle can recycle, continue again. Now, the very good news is you do not have to memorize every single little step along the way. We just want to know the basics of the 3 parts of this cycle. So let's take a look at the Calvin cycle. The first part is where carbon dioxide enters into the cycle. That carbon dioxide came from the air. It enters basically through this process 1 molecule at a time. Phase 1 is called carbon fixation. In carbon fixation, that carbon dioxide is fixed into a short-term intermediate molecule. We don't have to know all of that process, but we do need to know that that's done by an enzyme called rubisco. So the CO2 comes in, the carbon is attached to a 5-carbon sugar that's called RUBP, and that RUBP will be recreated at the end here. That is done by an enzyme called rubisco. And rubisco is very well known because it's thought to be the most abundant protein on the entire planet. It's created by plants at the rate of about 1,000 kilograms per second worldwide. So there are literally tons of this protein on our planet. And the question probably would be, why is there so much? Why do plants create so much Rubisco? And well, a couple of different reasons. One, because it's so important to this particular process, and also because it's kind of an evolutionary mechanism. And like many evolutionary mechanisms, they're not always the most efficient. They're not always the most well designed, because evolution can only use existing components to make changes or to create novel pathways. So you don't always get perhaps the most efficient design. It's thought that Rubisco was first evolved in a time when the atmosphere had very low oxygen. And now that it has very high oxygen, we're going to see later that that makes Rubisco sort of inefficient and problematic, but the plants are kind of stuck with it, at least for the moment, for now. The Crash Course video does a really good job talking about Rubisco and how it might be not the most efficient way for things to go, but like we said, evolution sometimes is a messy process. So step one is fixing the carbon from CO2 using Rubisco, attaching that carbon into a sugar molecule called RUBP. Step two, again, the good news, we don't have to remember all of this, but this is going to require the input of energy. Here are ATPs and NADPHs going into this process. Remember, those ATPs and NADPHs came from the light portion of photosynthesis that we already talked about. So this is called the reduction portion of the Calvin cycle. From this, we're going to end up with 6 G3Ps, but unfortunately 5 of them are going to be used in the last step. So we only get 1 net G3P, and then that can be used to eventually go on to combine with other G3P molecules and create glucose and other organic compounds. Then the final step is called regeneration, because we're going to have to get back to where we started. So through using those 5 G3Ps and again using more ATP, we're going to regenerate the RUBP that we started with, and then we'll be back to where we began, start the whole process over, more carbon dioxide, round and around we go. So altogether in this cycle, to get 1 G3P molecule, we're going to end up using 9 ATP and 6 NADPHs. All of those came from the light-dependent reactions that were part 1 of photosynthesis. So again, you don't have to remember every single little detail here. Just kind of remember the Calvin cycle is the second part of photosynthesis. This is where we're going to take carbon from CO2 and actually use the energy from the first part of photosynthesis to create eventually G3P, which will go on to create glucose and other food molecules. We'll take a break. We'll make this one a short one. We'll come back and talk about the last few minor things about photosynthesis and some of the different plants that do it a slightly different way, and we'll wrap it up